Hi, everybody, happy Friday. Welcome to this week's Single Cell and Exploration Club, where this week we'll be talking about glioblastoma, and I'll be using a case study on SmartBulk to go over the analysis today. But before we go over the talk today, I want to make a really important announcement to our schedule. So we'll still be hosting SCSE Club every Friday, except every other Friday. Starting from next week, we'll be hosting SCSE Club starting at 9 a.m. CEST or 4 p.m. JST. So this is to we want to involve our users from Europe and Asia a little bit more. So that's why starting next Friday, June 13th, and every other Friday following next week, we'll be hosting during Europe time. And then for Friday, June 20th, we'll go back to our regular schedule time and every other following Friday from that, 9 a.m. PST or 12 p.m. EST. Okay, so I'll be making this announcement again at the end of SCSC Club for anyone who misses it. So before I go over the agenda today, this is the feature that I'm going to demonstrate today in our club. This is the cell type deconvolution feature in SmartBulk. So this is what helps us get the cell type proportions for each of our samples. If we start from a bulk RNA seq matrix and we include a gene signature matrix that is generated from a single cell RNA sequencing data set, then we can get the cell type proportions within our bulk data set. Okay, so jumping into the agenda today, I'll be going over the genes and the pathways that are upregulated in glioblastomas. And we can also find out the cell type proportion differences in glioblastoma versus normal. And then finally, I was able also to identify a potential high-grade glioblastoma subtype as well. Okay, so let's get started about talk a little bit about what glioblastoma is. So gliomas are the umbrella term for glioblastomas. These are brain tumors originating from glial cells, and glial cells are sort of the supportive cells for neurons, but they also play an important role in central nervous system function as well. Glioblastoma is known as the most common and aggressive type of brain tumor. And the term glioblastoma itself is classified as a high-grade tumor, whereas if you refer to a low-grade tumor, you would call that a low-grade glioma. Some important cell types to keep in mind is the glioblastoma stem cell, or the GSC. These stem cells are highly resistant to cancer treatments, mostly because of their plasticity and their ability to evade treatments and whatnot. So this is one of the reasons why I wanted to investigate the heterogeneity in glioblastoma. For the data set that we'll be using today, it comes from this paper, Diversity and Divergence of the Glioma Infiltrating T-Cell Receptor Repertoire. So in this paper, the authors have also performed a TCR-seq as well. But for the purposes of today's demonstration, I'll only be going over the bulk RNA sequencing data set where they took samples from brain tissue. You can find this data set on SmartBulk Public Studies by searching for or by filtering for the brain, and then you can find it with the study name and study ID down below. Okay, so one of the first analysis that I wanted to do was kind of characterize the differences between high-grade and low-grade tumors. So I performed two different differential expression analyses, one comparing high-grade versus normal, and then one comparing the low-grade versus normal. And what you're seeing here is a cropped version of the volcano plot, which is generated from the differential expression analysis, because I wanted to see what genes were upregulated in our high-grade tumor. So on the left-hand side, we see some upregulation in marker genes of the glioblastoma stem cells, such as CD44, NES, S100A4, SOX2, and CD70. You can also see the enrichment here on the volcano plot. Whereas on the right-hand side, where we compare our low grade versus our normal, I don't see that upregulation of these marker genes of glioblastoma stem cells. So what this is telling me is that these low-grade gliomas don't yet express many marker genes of our stem cells. However, an interesting pattern I do see is the upregulation of this SAA1 gene. I see that upregulation in both the high-grade and the low-grade tumor. But an interesting pattern that I see is that this SAA expression may correlate with the grade of the tumor, where you see in the low grade, the SAA1 is a little bit less upregulated compared to our high grade, in which it's very, very upregulated. Some studies have also reported that this SAA1 gene has been implicated in poor prognosis as well. So it's interesting to see this pattern play across in bulk RNA sequencing data. Next, since I've identified some of the genes that are upregulated in our high-grade and low-grade tumors, I also wanted to see what pathways were enriched in my diseased groups too. So what you're looking at here is a GC plot or a gene set enrichment analysis plot, where each of these colored lines, the green, the purple, and the orange, represent one single pathway. And when you see the pathway trend going down, towards the right-hand side of the graph where you see more enrichment in the downregulated genes. So you can interpret this graph in that all of these pathways, these three pathways here, are downregulated in our high-grade tumor. The pathways are related to some neuron functions, such as neurotransmitters secretion, positive relation of synaptic transmission and neuronal system. So overall, I see the same pattern across my high-grade tumor and my low-grade tumor. So overall, I see a downregulation of neuron communication across both grades of the gliomas. Okay, next, since I performed two different analyses for differential expression analysis, comparing high grade and low grade, I also wanted to see what kind of genes overlapped within our two lists of differentially expressed genes. So I can do that by using this Venn diagram analysis here, where I'm comparing the differentially expressed genes that are upregulated in my high grade tumor with my differentially expressed genes upregulated in my low-grade tumor, and I see that there are 188 overlapping differentially expressed genes. And when I perform a pathway analysis on this 188 genes, I can see an overall upregulation in cell cycle and cell differentiation pathways across low-grade glioma and high-grade glioma or glioblastoma as well. So what this is telling me is that there is a lot of high proliferation or a lot of cell cycling going on. So basically a lot of cell growth in the tumors that we're seeing. Okay, so next we've arrived to our cell type deconvolution. Again, I wanted to see within each of my samples these cell type proportions. So I'm going to go ahead and demonstrate this on SmartBulk here. So in SmartBulk, you can find the cell type deconvolution feature here on the left-hand side. You'll click it. So we'll name the analysis deconvolution. And for the data set that I'll use, I want to use my normalized data set. And from the references, you can choose between our two publicly available references that we have, one from this study and one from this MCA3 study. For this data set, I chose the MCA brain. So I'll choose that as the reference. So this reference is the gene signature matrix that was provided from this particular study. I'll click Run Analysis, and the results page will look like what you're seeing here. So for each sample on the x-axis, you'll be able to see all of the different cell types within that sample. The plot is also interactive, so if you hover over one of the bars, you can see the cell type fraction or the cell type portion. You can also order this graph as well. So for example, if I want to order by the samples, I'll go ahead and click apply, and then I can also show the grouping or the metadata of the samples. So I want to see how my samples are grouped into the normal versus the low grade versus the high grade tumor. I can click the settings button here, which will open up all of these settings to configure the stacked bar plot. And for the metadata, I will choose the condition. Let's see, the condition subtype. Okay, so this is actually, I'm getting a little bit ahead of myself where the subtypes refer to the high grade tumors. So you can see here on the left-hand side, these three are our normal samples. Next to that are the low-grade glioma samples, and then following that are all of the glioblastoma or high-grade tumor samples. One pattern you can notice right away is that for both our normal and our low-grade tumors, they're very, very high in astrocytes, but not many other types of cells. Whereas in our high-grade tumor, you can see a lot of heterogeneity in cell type proportion, and an overall pattern of decreasing astrocytes as well. In order to get the statistical significance of whether this decrease in astrocytes that I'm seeing is actually, statistically significant, I can go over here down to the cell type comparison, click this open and create a new comparison analysis. So for this comparison, I want to compare my high grade versus my, excuse me, my high grade versus my normal. Mm. So there's two ways I can perform it, either by selecting by the experimental group or by the composition analysis as well. So that's what I'll do here. Okay, for my group 1, I will name this high grade. And I'll choose the subtypes, which are the high grade. And for my group 2, I'll choose that. And click Run Analysis. So the results from running this analysis will give you a box plot with the statistical calculation. So for T cells, you can see it's not really significant, but if we go down to our astrocytes, this is where you can really see the differences. So for our normal, you see a higher cell type fraction in astrocytes versus our high grade, you can see that decrease. And that this difference here is also statistically significant, which we represent with using these asterisks. Okay, so again, to summarize, I saw this pattern of where the high-grade glioblastomas exhibit more cell type variation and more differentiation in the cell type proportions. And when we looked at the comparison between our normal versus our high grade in our astrocytes, we can see a marked decrease in astrocytes in our high grade tumor. Okay. Next, I also want to highlight a new feature that we released in SmartBulk, or a new enhancement to the cell type deconvolution, which is where not only can you perform cell type deconvolution using the public signature matrices that we provide for you, but if you also have your own single cell data set that you want to use as a reference for your cell type deconvolution, you're able to do that as well. So within the deconvolution tool, down here, you can click custom references, and this is where you can upload your own signature matrix that was generated from your single cell RNA sequencing data into the platform here. So for example, I have a matrix that I saved on my computer. I can easily drag and drop that, and it will populate it here. And you can choose that as a reference to run the analysis. So currently, we are supporting users in creating custom signature matrices for them. So if you're interested in performing cell type deconvolution using your own custom signature matrix, you can always contact us at support@bopturing.com, and we'll be able to generate that custom signature matrix for you so that you can use it in your deconvolution. So the results from that custom signature matrix that I uploaded, the results I see are a little bit different. However, overall, the pattern that I do see is that there's still a lot more cell heterogeneity within our tumors. So I used the custom signature matrix from this particular study. So again, overall, the pattern that I see in both low and high grade glioma is a lot more different cell types compared to our normal, at least. Okay, so another question I can ask from here is, so I want to see what kind of genes are kind of co-expressed with each other and what kind of, what that co-expression could be associated with. And so this is where I can use the tool called weighted gene co-expression network analysis, or WGCNA, to find out which gene modules are associated with the kind of traits or the kind of phenotypes that I have. So basically what WGCNA does is it sees what kind of genes are co-expressed with each other, and it groups those co-expressed genes into different modules. And with those modules, you can also associate them with a known phenotype or a known trait um, from your data set, and see what kind of modules and what kind of hub genes you can identify from each of those modules. So from the WGCNA analysis, it was able to identify 4 different modules. And when I associated it with the condition, I noticed that module 1 was very highly associated with my high-grade tumor. And if I were to look into some of the hub genes for module 1, you can see them over here listed on the right-hand side. These are the top 10 hub genes. Hub genes, you can kind of interpret them as very highly interconnected genes within this certain module. So these genes are most likely the drivers for this particular module. And another thing that I noticed is not only is the high-grade tumor associated with module 1, but also the low-grade glioma and the normal conditions are associated with module 4. You can see that here. So we saw this pattern of the low-grade glioma and the normal conditions kind of associating with each other as well, because when I performed the cell type deconvolution, we also saw that they had similar cell type composition. So going back a few slides here, you can see here our normal and our low-grade, they have very similar cell type composition. Okay. And so next, now that we've identified that possibly our low grade and our normal conditions might be associated with each other, I wanted to dig a little bit deeper into our high grade population. So this is where I use the single sample gene set enrichment analysis within my high grade tumor population. So this single sample gene set enrichment analysis allows me to see what kind of pathways are enriched within each of my individual samples. So the heat map that you're looking at here is on the top x-axis is each individual sample, as well as the condition that they're assigned. And on the y-axis are the individual pathways that are enriched within each sample. And you can kind of see an overall pattern going on within this second cluster on the right-hand side here, where the samples that are normal versus low grade, they're downregulated within this section of the pathways. However, within my high grade population, I kind of see some heterogeneity going on where some samples that are high grade tumors are upregulated in a pathway, but other samples are downregulated in those pathways. So this leads me to believe that there are possible subtypes even within our high grade population. So taking a subset of the selection that I've boxed here, I was able to subset this selection and recluster them to get a new heat map, which you see here. So again, what this heat map identified is it identified two particular clusters, the orange and the blue. And you can see within the orange cluster, those pathways are downregulated, and within our blue cluster, the pathways are mostly upregulated. And the pathways that we're looking at here are mostly related to pro-inflammatory pathways. So naming off a few of them, you can see IL-1 and megakaryocytes, or inflammatory bowel disease signatures, or B cell receptor signaling. One that I've highlighted here on the right-hand side is the IL-11 signaling. So typically this is a pro-inflammatory pathway. However, IL-11 has been implicated in kind of promoting glioblastoma proliferation as well. So when we look at the pathway enrichment between our two different subtypes, you can see that there is a downregulation of IL-11 in particularly our subtype A or our orange subtype. So this leads me to believe that there's this subtype right here is kind of suppressing this IL-11 signaling. And this actually might be a good thing because if IL-11 has been implicated to kind of further progress glioblastoma, then maybe a suppression of this IL-11 could lead to a better or a more positive response to cancer treatments. Okay. Another way I kind of want to assess that hypothesis, whether these subtypes may be responsive to treatment or not, is by using our new tool in SmartBulk that's called sample clustering. So in sample clustering, what it does is it will cluster together your samples based on how similar their gene expression profiles are. And this is especially useful if you don't really have any metadata associated with your samples, or if you want to see, for example, I know that my samples have different conditions assigned to it. This is based off of, some histopathology diagnosis or some imaging. We know that some of these samples are high-grade tumor versus low-grade tumor, but we also want to further look into the gene expression and whether these are actually high-grade tumors or low-grade tumors, gene expression-wise. And so this is one of the ways that this sample clustering tool can help us. So what you're seeing here is, again, each column represents a different sample, whereas the rows will represent different genes, and then those genes are also clustered into an overall pathway as well. Okay, again, what we see here is the sample clustering has identified 3 different clusters. Okay, and within those clusters, you can see the first cluster here is mostly composed of our subtype B, uh, subtype B samples, whereas cluster 2 is mostly composed of our normal, versus cluster 3 has a mix of our subtype A plus our low-grade glioma as well. So it's really interesting to see how one of these subtypes of our high-grade tumor cluster together with our low-grade glioma. And so this also leads me to believe that since this particular subtype A clustered together with our low-grade glioma, possibly this subtype A may be more responsive to treatment. Okay, and so for the summary of today's talk, we were able to look at how our high-grade glioma samples exhibit upregulation in glioblastoma stem cell marker genes and cell cycle pathways. We also saw how normal and low-grade glioma samples display similar cell type proportions and associate with the same gene module. And performing single sample gene set enrichment analysis, it was able to reveal 2 subtypes of high-grade glioma, one of which is immunosuppressive. So we saw that in our subtype A, where it was kind of downregulated in the IL-11 pathway. And then lastly, we can also make the hypothesis that the immunosuppressive subtype may be more responsive to treatment, especially when we saw the clustering with the low-grade glioma. Okay, so that concludes today's talk. Again, I want to make the announcement for this schedule change to SCSC Club. So again, we are still hosting SCSC Club every Friday, but starting next week and every other Friday following June 13th, we'll be hosting SCSC Club at 9 a.m. CEST or 4 p.m. JST. And then starting Friday, June 20th, and every other Friday following that, we'll be hosting it at 9 a.m. PST and 12 p.m. EST. So at this time, my colleague will open up the poll for everyone to answer, and then I can also answer any Q&As as well. Okay. Yeah, my colleague has just opened up the poll for you guys. So we definitely want to hear your feedback on SmartBulk, especially for the cell type deconvolution feature that I demonstrated today. Interested in whether you guys are interested in us having more publicly available signature matrices, or if you were just more interested in uploading your own custom signature matrices as well. And for any of the tools that, or any of the features that I mentioned today that were hard to understand, during the next SCSC Club, whenever we present SmartBulk, we'll be sure to go over that in a little bit more depth. Okay, I'll give it a couple more seconds for you guys to finish answering your polls and sending in any questions you have. Okay, so I think that's good. So thank you everyone for joining today's SCSC Club, and we hope to see some European users and Asian users next week. If not, please tune in on June 20th, where we will have the SCSC Club at 9 a.m. PST and 12 p.m. EST. Thank you, everyone.